So I have entitled my talk, Early Life Lessons from the Microbiome. And I have a few disclaimers to make. Firstly, I am not an immunologist. And I think immunology is fascinating. I think it's really hard to understand a lot of the time. It makes my brain hurt sometimes, but I think it's really a fascinating area. And so, like I say, I'm not an immunologist, but I am interested in it, and I'm going to try my best. Also, I'm not a microbiomeologist. I think that anybody who thinks they are a microbiomeologist at the moment is probably misguided. I think there are certainly people who know more than others, but I think it's an area where we lack understanding more than we have understanding. So this is a little bit of a cook's tour. I'll admit to having gone down some rabbit holes when I was preparing this talk, but I hope that some of you will find it interesting. So obviously the microbiome has become a very hot topic in the last few years. And we are starting to realize that we are much more closely connected to the environment than we thought, around us, within us, and that we are much less the higher organisms than we think we are. We and the organisms that we carry are integral to each other's survival. Of course, the organisms that we carry would quite happily find other modes of transport if we were not available, and they do certainly find other things to do when we expire on them, but we are much less well equipped to do without them, and even poorly equipped to cope when the ratios of those organisms are slightly disturbed. So these days, you can't really be sure that any of the diseases that you treat aren't related in some way to the function or dysfunction of the microbiome. So firstly, some definitions. The definition of the microbiome itself is basically a community of organisms, bacteria, viruses, archaea, fungi, and their genes that are present in a particular environment. So when we talk about the microbiome, we talk about the organisms, and the term incorporates the environment in which they exist as well. And in our case, that environment is obviously the human body. These organisms may be commensal, symbiotic, or pathogenic. And the balance between helpful and harmful organisms is generally maintained in a healthy person. The microbiota is the term used to describe only the organisms themselves. The metagenome is the collection of genomes and genes from the microbiota. The metatranscriptome is the collection of all expressed RNA in the specimen. And the proteome is the entire protein complement in the sample. And the metabolome is the entire metabolite profile in the sample. And together, those two things give an idea of the regulation and expression profile and function of the microbiome. And there are anatomical sites where it is very well accepted that a microbiome is present, sites where organisms are expected to be and which engage in complex interactions with the host. The greatest number of organisms with the greatest diversity exists in the gastrointestinal tract, and of those, the most reside in the large bowel. And the disclaimer here is that a lot of the discussion today is going to focus on this large and important component of our overall microbiome. Other sites obviously include the skin, also a source of many and varied organisms, the orifices of the head, the conjunctiva, the genitourinary system, the lung, and the male and female reproductive tracts. Also, just to mention that it's not true that we have 10 times as many bacterial cells in our bodies than we do human cells. It's probably closer to 1-to-1 or maybe 1.3-to-1, depending on how many times you've gone to the toilet in the last little while. But without doubt, we carry a teeming civilization of organisms around with us. And one can never say never when it comes to a microbiome. It's an area that's always evolving, and understanding is evolving. But it's probably fair to say that at present, there is little evidence for a microbiome in the brain, in the heart, in the liver, the spleen, and the bloodstream. But there are researchers who have described what they believe is the microbiome of the brain, although from what I've read, these microbiomes that are described do seem to be associated with disease. So I'm not sure what constitutes a microbiome, but maybe we are all just zombies controlled by our brain microbiota. And as far as emerging microbiomes go, in particular, I'll discuss a little later, one of the more recent microbiomes postulated to exist, which is that of the fetal environment. Now, none of us, I think, would dispute the presence of a uterine microbiome. It does, after all, communicate with the outside world, and that certainly leads, promotes the establishment of a microbiome. But a fetal microbiome is maybe still uncertain. So firstly, how do we go about measuring a microbiome? So the earliest microbiome studies involved the traditional techniques used in microbiology. So basically culture of a specimen to grow a colony of organisms. This works well for bacteria and fungi, not so easy with viruses. And then you would identify the organism in that culture by morphology and biochemistry. And the microbiome may only have become dinner table conversation in the last decade or so, but it's been of interest to some for much, much longer. For example, Theodore Escherich, whose name we will all recognize, I'm sure, cultured the meconium of newborns as early as 1885 to see what, if anything, grew in the stools, in the first stools of life. And then in 1927 and 1934, two other studies also examined meconium of neonates by collecting it in sterile nappies and then attempting to culture it. Escherich found no viable bacteria, and the other two studies found that 2/3 of samples didn't culture anything. But these days, obviously, culture-based technologies are not used to study microbiota for several reasons. So firstly, many of the bacteria colonizing our guts are obligate anaerobes, which often don't survive the procedures that we use to sample from the GIT, and they also don't necessarily survive transport to the laboratory and storage. Also, the bacteria found in the gut grow with variable efficiency in the laboratory. The organisms that would be most readily isolated would be those that grow under aerobic conditions at moderate temperatures and with high nutrient growth media. And the bacteria that meet those criteria probably only make up 1 to 2% of the sample that you're actually interested in. And I came across the term, which I really liked, which is that these organisms represent the weeds of the microbial world. And so they can't be said to represent the numbers or the variety of microbes that constitute the gut microbiome. So at the moment, culture would not be regarded as a very useful tool for assessing the composition of a microbiome. So what are the alternatives to culture? Well, all living cells contain ribosomes. And I had to remind myself that this is the organelle that turns messenger RNA into protein. And ribosomes are composed of two subunits, one a large subunit, the other a small subunit. And the small subunit contains an RNA molecule, which is referred to by its size as either an 18S in the case of a eukaryotic cell or 16S in the case of a prokaryotic cell. The 16S subunit is encoded for by the bacterial genome. The gene that encodes the 16S ribosomal RNA is both highly conserved, it has highly conserved sequences, and hypervariable regions that can then be used to identify and classify bacterial species. And the current sequencing techniques, this is bacterial 16S ribosomal RNA is currently regarded as the gold standard for assessment by molecular or nucleic acid sequencing techniques. And current sequencing techniques allow sequencing of all the DNA molecules that are present in a DNA mixture at the same time. For example, the mixture of DNA molecules that would be obtained from a stool sample, which we would expect to contain DNA from many, many different sources. So the PCR method would involve nucleic acid amplification of the whole 16S gene or portions of the gene which contains the hypervariable regions, by which you would be able to speciate the organisms. And this is done by techniques like gel electrophoresis, fish flow cytometry, or DNA microarrays to separate the fragments of different length. So the problem with PCR is that these techniques are not sensitive enough or are not always sensitive enough to distinguish between some bacterial species. And sometimes you may not even be able to distinguish between genera of bacteria or families or orders. And another problem with this method might be that bacterial species occurring in lower abundance may not amplify and therefore may not be detected. So again, you may well be picking up the weeds of the microbial world. But whether we can always effectively detect or characterize our microbiota, it is very clear that these organisms are integral to normal functioning in the human host. And that influence begins very early at life. So just some background on the neonatal immune system. The neonatal immune system is understandably immature. It has essentially no immunological memory, which is why neonates rely on maternal antibodies and to a large extent, the innate arm of the immune system for protection from infectious diseases in the first few months. Maternal antibodies wane over about 6 months, give or take. Some last longer than others. And the components of the neonatal innate immune system, which are functional, are the following. So they have cellular components, which includes dendritic cells, neutrophils, macrophages, monocytes, and natural killer cells. And extracellular components, which include complement, fibronectin, antimicrobial peptides and proteins, and acute phase proteins. But just because they are present doesn't mean they work all that well. So the neonatal immune system is deficient in a number of ways. So firstly, neutrophils are deficient in both quantity and quality. They have lower expression of toll-like receptor 4. They have deficient downstream signaling of MyD88, which ultimately results in the inhibition of tumor necrosis factor alpha secretion. They have lower expression of adhesion molecules, so their neutrophils adhere poorly and they transmigrate poorly. They have deficient chemotaxis in response to chemotactic factors. They are deficient in making neutrophil extracellular traps for killing extracellular organisms. They have reduced phagocytic ability, and they have defective NADPH oxidase systems with reduced generation of hydroxyl radicals. So they have neutrophils, but they really don't work all that well. What about antigen-presenting cells? So they have fewer numbers, so they have fewer dendritic cells and fewer monocytes. And those dendritic cells and monocytes express reduced levels of MHC class II molecules, CD80 and CD86, which basically makes them inefficient activators of antigen-specific T and B cell responses. Their toll-like receptors tend to induce an anti-inflammatory cytokine response. So IL-10, IL-6, and IL-23. This changes over the course of about a year to become a more pro-inflammatory IL-1 beta phenotype. Natural killer cells, they actually have more natural killer cells. Natural killer cells play a major role in the resolution of severe acute respiratory viral infections like influenza and RSV. But these cells are more of a CD56 bright subset, and that particular subset is poorly cytotoxic. They express high levels of cytokines and they home to lymph nodes. And they express higher numbers of inhibitory receptors. They are also have a reduced ability to lyse targets without undergoing differentiation. So undifferentiated cells are not as good as lysing targets, but they have similar antibody-dependent cellular cytotoxicity to adult cells. So what about gamma delta T cells? So gamma delta T cells are first responders to microbial infections like Mycobacterium tuberculosis, Listeria, and Brucella. And in adults, they constitute about 1 to 5% of the lymphocytes in peripheral blood. In neonates, they have a lower percentage. So there's a lower percentage of gamma delta T cells in human cord blood. Those that are present in cord blood have a naive phenotype. They are poorly proliferative and they have reduced cytokine responses. They are weakly cytolytic and they have reduced perforin and granzyme B effector molecules. So what about the adaptive immune system? The adaptive immune system in neonates also demonstrates both qualitative and quantitative deficiencies, and this persists until about the age of 18 months. So T cells tend to be polarized towards Th2 responses. They tend to produce more IL-4, IL-5, and IL-10, with decreased production of Th1 cytokines, so interferon gamma, IL-2, and tumor necrosis alpha. Suppression of interferon gamma secretion by Th1 cells was shown to be due to higher expressions and secretion of IL-4. It's been demonstrated that skewing of naive T cells towards a Th17 or Treg cell is mutually exclusive. So these naive T cells tend to become either Th17 or Treg cells, one or the other. They can't kind of be both or revert. And if you have naive T cells that are in the presence of TGF beta and the presence of IL-6 and IL-21, they tend to become Th17. If they're in the presence of TGF beta and IL-2, they tend to become FOXP3 positive Treg cells. And obviously, Treg cells are designed to maintain immunological self-tolerance, and they negatively regulate various immune responses. So they downregulate pro-inflammatory immune responses. Th17 cells, there is a low frequency or an absence of Th17 cells in cord blood. Th17 cells are typically pro-inflammatory, and it kind of makes sense that they're basically the opposite of Tregs. But in fact, in neonatal immune development, they may play a slightly different role. They aren't typically pro-inflammatory. The innate cells differ to adults in the activation-induced cytokine production. So the activated cell cytokine profile reduces the upregulation of Th17 transcription factor. And Th17 cells may in fact help to prevent adverse reactions or inflammation in response to microbial commensals. So typically we think of Th17 cells as pro-inflammatory, but in this situation, they may in fact promote some degree of tolerance to microbial commensals. All right, so Tregs are present in high frequency in cord blood and in neonatal lymphocytes. That neonatal lymph nodes. That kind of makes sense because they have just been delivered from a very tolerogenic environment. The fetus tolerates the mother and the mother tolerates the fetus. As I mentioned, they are the reciprocal cell of Th17 cells. And neonatal non-differentiated T cells preferentially differentiate into Tregs in response to stimulation of the T cell receptor. CD8 cells are reduced in number and function, and their CD8 activation is impaired, but they do demonstrate an adult-like response to viral infections and vaccines. B cells, naive B cells, they've had no prior antigenic exposure. They have a partially developed surface immunoglobulin repertoire. They have reduced strength of BCR signaling, a higher expression of negative regulators of B cell receptor signaling and cross-linking of antigens to the B cell receptor, and they have defects in maturation and B cell isotype switching. So essentially what it boils down to is an overall tolerogenic environment, particularly at mucosal sites, with a unique response to microbial-associated molecular patterns, which promotes essentially healthy microbial colonization. The fact that our immune systems are so deficient, it to me is a miracle that any of us make it out of infancy alive. And even more amazing when you consider that we are immediately invaded by a multitude of organisms that will colonize our skin, our mouths, our lungs, and gut pretty much before we've even let out our first scream. And most interesting, that we require that interaction with those organisms to develop a more effective and mature immune system. So how does that happen? So let's think about what happens if you prevent colonization of an animal altogether. So this is the mouse in a bubble idea. These are referred to as germ-free animals, which are bred and raised to be entirely sterile. And basically, these animals fail to develop normal immune function and immune anatomy at the mucosa and systemically. So firstly, they have grossly abnormal architecture of the gullet, the spleen, and the thymus. And we must remember that the gullet is one of the largest organisms of the immune system in the body. It includes the Peyer's patches, which house dendritic cells and neutrophils and macrophages. It includes the mesenteric lymph nodes. It includes lymphocytes that are scattered throughout the intestinal epithelium as well. And in fact, about 80% of immune cells reside in the intestine in adults. So germ-free animals have they have reduced, in addition to abnormal architecture, they have reduced frequencies of CD4 and CD8 T cell subsets. They have reduced numbers of lymphocytes in the intestinal epithelium, and they have altered T cell cytokine polarization with a lack of diversity at the alpha beta T cell receptor, at the T cell receptor. If you introduce microbiota into the GIT of germ-free mice, it in fact restores those defects. In particular, if you introduce Bacteroides fragilis, which has been shown to induce IL-10 producing Treg populations and to limit pro-inflammatory IL-17 responses, Clostridial species also promote Treg development. And then segmented filamentous bacteria, which are gram-positive anaerobes, promote the accumulation of Th17 and Th1 cells in the small intestine. And that accumulation of those cells drives the production of IgA, which obviously is a significant component of mucosal immunity. So in germ-free animals as well, B-cell maturation is in fact normal, but there are defects in the production of IgA and IgG1, both at the mucosa and systemically. And IgA functions, the function of IgA is to compartmentalize intestinal bacteria and to prevent the adherence of those bacteria to intestinal cells, which could potentially be a precursor to invasion. So dendritic cells sample commensals in the lumen. They then interact with T cells and B cells in the lymphoid tissues. And then B cells that are producing IgA migrate to the intestinal lamina propria and secrete the IgA, which is then transcytosed into the epithelium. Also, there's a new class of innate cells called the innate lymphoid cells. These are lymphoid cells that have no T cell or B cell receptors, but they still function in a cytotoxic and immunoregulatory fashion. They mimic the pattern of T cell cytokine secretion, but in an antigen non-specific way. And there are two groups that we are interested in. So group 1 cells, which includes NK cells. These are hyperactivated in germ-free animals. And they respond to inflammatory signals from non-mucosal phagocytes, which in normal animals which are not germ-free, those inflammatory signals are usually restricted by the presence of those commensal bacteria. And then group 3 innate lymphoid cells secrete less IL-22 in germ-free animals. IL-22 usually promotes production of antimicrobial peptides. It stimulates regeneration of the mucosa. It increases mucus production at the mucosa, and it regulates wound repair. And then intestinal epithelial cells in germ-free animals, there is altered microvilli formation and lower rates of cell turnover. So now we know that neonates have barely functional immune systems. We know that we need our microbiota to educate our immune systems to become appropriately tolerogenic and inflammatory. But the question remains, when does that interaction actually begin? So when do we acquire our microbiota? So firstly, we think, and many of us are aware of this, that the mode of delivery impacts the baseline composition of your microbiota. So during vaginal delivery, the neonate is more likely to acquire organisms like Bacteroides, Bifidobacterium, Lactobacillus, and Provotella species. During cesarean section, as the neonate isn't in direct contact with maternal, vaginal, or fecal microbes, that neonate is more likely to be colonized by skin or environmental flora, including those from the hospital staff and the hospital environment, which we know are awful, awful bacteria, more and more every day. such as propionine bacteria, corynebacteria, streptococcal species, and, God forbid, all sorts of other nasty things. And these and babies that are delivered by cesarean section are also more likely to acquire a greater number of Clostridial species. However, regardless of the mode of delivery, very shortly after birth, the neonate is going to start to acquire organisms from the skin of the mother and from other individuals, from the mother's breast, from the organisms present in colostrum, and the organisms present in the environment. This variation between babies born by normal vaginal delivery and by cesarean section decreases with time. And there are studies that have shown very variable duration of persistence of this difference. So it's been reported as anything between 1 month, so essentially just the neonatal period, and then organisms even out, they look much the same, and 7 years. So there is the suggestion that cesarean section may be associated with long-term health consequences, which may in turn, although this is not clear, be related to the different ways in which the microbiome is established. And these long-term health consequences include things like altered cytokine expression, susceptibility to immune disorders like asthma and allergy, type 1 diabetes, and an association with obesity. But obviously these associations are very hard to tease out because these are lifelong conditions. These are things that may present only in adulthood sometimes. And there are multiple other factors that may influence the development and maturation of the microbiota, such as feeding mode, breastfeeding versus formula use, diet in childhood or adulthood, maternal age, the metabolic status of the mother, obviously your genetics, the lifestyle that you lead, and then, of course, antibiotic usage as well is going to affect your microbiome. But recently, the idea of the sterile womb, which got its start in the days of Theodore Escherich, who was culturing meconium. So he said there's nothing in meconium, and so the womb must be sterile. And this idea has been challenged. So when I say the sterile womb, obviously I mean the sterile fetal environment rather than the sterile uterus. And so if the fetal environment is not sterile, then the idea that the mode of delivery impacts significantly on the composition of the microbiome must also be challenged. And this is very controversial. I don't know quite which side of the fence I sit on. That may become clearer just now. But with the advent of the molecular techniques for detecting and characterizing the microbiome, bacterial DNA has now been detected in placental tissue. It's been detected in amniotic fluid. It's been detected in the umbilical cord and detected in what seem to be healthy full-term pregnancies. So what traditionally do we think protects the fetal environment from infection? And if those barriers can protect against infection, why don't they protect against colonization? So both of which would essentially constitute invasion, but just of different degrees. So the maternal fetal barrier consists of two anatomically distinct elements. It consists of the chorioallantoic placenta and the chorioamnion. And this barrier is formed at the placental level by the villus syncytiotrophoblasts, which is a layer of specialized epithelial cells that differentiate from cytotrophoblasts. The syncytiotrophoblast actively invades the uterine wall and eventually forms the outermost fetal constituent of the placenta and the placental villi. And I must say, I have great difficulty kind of picturing in my head the invasion without mingling of the tissues. But essentially, the syncytiotrophoblast acts as a continuous cell without any intercellular barriers. And so that prevents both maternal and bacterial cells from squeezing through any intercellular junctions that have to go directly through what is essentially one long cell and then into the fetal bloodstream. And so this is the first level of structural protection against the invasion, both of maternal cells, which might be carrying foreign antigens or potentially destructive immune cells, and bacterial pathogens as well. And then there's a basement membrane that separates the syncytial trophoblast from the connective tissue, which contains the fetal capillaries. And then there are what's known as extravillous trophoblasts, which invade the decidua, which is the maternal part of the placenta. It functions to anchor the placenta into the uterine wall. And those extravillous trophoblasts, they exist in the same environment as natural killer cells and macrophages and leukocytes, and they together provide some innate defense mechanisms, and they possess some intrinsic bactericidal properties. So together, these are the physical barrier that prevents the passage of bacteria into the amniotic sac and then consequently into the fetus. Obviously, it's not foolproof. Obviously, infection of the placenta and the fetus does occur. So, for example, with Listeria or Brucellus or Toxoplasma, and I know Jill did a long discussion recently about maternal and fetal infections. And those organisms in particular, we think, possess factors that allow successful invasion across those barriers. So, for example, Listeria has internalins, it has Listeria lysine and various other things which allow it to cross not only the placental barrier, but also, for example, the blood-brain barrier. So there are specific organisms that we know are able to subvert these placental defenses. So we know that it's not, it's obviously not 100% protective. So what about immune cells at that placental barrier? The immune cells provide protection not only to the fetus from infection, but they prevent the exchange to some degree of antigen and protection of the mother from the fetal antigens and vice versa. And the cells that exist there are natural killer cells, regulatory T cells, macrophages, dendritic cells, and all of those cells are necessary for a successful pregnancy. The placenta itself also expresses toll-like receptors and nod-like receptors that provide a protective response against pathogens and against danger signals. And those receptors are differentially regulated depending on the stage of gestation. Also the maternal reproductive tract expresses antimicrobial peptides, which provide a chemical barrier against ascending infection, and those increase late in gestation, and some of those peptides are even released directly into the amniotic fluid and into the fetal compartment during labor. Also, on the maternal side of the placenta exist immunoglobulins, IgG, IgA, IgM, and those additionally protect against bacterial invasion. Right, so if in utero colonization occurs, this would require dissemination of maternal microbes either by the ascending route from the reproductive tract, and then we would expect the organisms that we mentioned earlier, lactobacilli, prevotella, etc., or by the hematogenous route from other maternal sites which harbor microbiota. So specifically, we're thinking about the mouth and the rest of the GIT. So then obviously we're saying that bacteremia with viable commensal organisms from these sites must occur. And this probably does happen. We probably develop transient bacteremias when we have dental procedures or even just brush our teeth. So hematogenous seeding is not outside the realm of possibility. And potentially immune cells might actually carry organisms from the mouth or the GIT to the placenta in order to facilitate an in utero establishment of a fetal microbiome, if that is something that occurs. So when do we find amniotic fluid being positive for organisms? So most of the time, it is not possible to detect organisms by any method. So not by culture, not by PCR or sequencing, or a combination of all of those methods. It is much more likely to be positive under pathological conditions. So organisms are detected in 15% of cases of preterm labor, in 50% of preterm premature rupture of membranes, in 10% of cases of preeclampsia, in 57% of cases of neonatal sepsis, which certainly makes sense, and in 6% of pregnancies with small babies, which suggests that the presence of organisms in the fetal environment is associated with poor pregnancy outcomes. Also, the duration of labor is associated with the detection of organisms in meconium. So if it's under 6 hours, meconium is more likely to be sterile. If it's over 6 hours, it's mostly culture positive, which suggests ascending colonization given enough time. And a phrase that I also particularly like, another phrase, is that infection does not a microbiome make. And if the presence of organisms is associated with outcomes, then they're probably not actually commensals, but rather would be classified as invaders. All right, so what about organisms actually in the placenta? So the study that really got people talking about the placental microbiome was a 2014 study by Agard et al., which determined by metagenomic sequencing that a unique microbiome existed in placentas from healthy, full-term and preterm, cesarean and normal vaginal deliveries, and that the pattern shared a microbial pattern with the oral microbiota. So here we're looking at the oral microbiota. Here's supragingival plaque, the tongue, the buccal mucosa, and this is the placenta. And then there's this little group of organisms, this little pink group of organisms here, which doesn't appear in the mouth. So that's the microbiota of the placenta is dominated by the proteobacteria, and then it also includes this phyla not found at other sites, which is the tenericutes, and that includes pathogens like ureaplasma and mycoplasma, which are associated with the uterine environment, but they are not considered normal vaginal commensals. And those, ureaplasma and mycoplasma are associated with preterm delivery and abortion. Also, the microbial patterns were not similar to the vaginal flora, which you would expect to colonize the placenta during normal vaginal delivery. So this is the vaginal microbiome, and you can see that it doesn't show the same. It doesn't show the same pattern. So organisms in this study were detected in low abundance. They were detected more often in preterm spontaneous births and in association with a remote history of infection during pregnancy. So by which they mean, when they talk about remote history of infection, they mean things like UTIs in the first trimester, which not only adds the confounder of, potential bacteremia associated with infections and perhaps dissemination then, but also adds the potential confounder of antibiotic exposure. This bubble plot here on the right-hand side shows that the most prevalent single species in the placenta in this study was E. coli. There we go. And I must confess that my gut response, pardon the pun, is that this is quite suspicious to me for contaminated or otherwise inaccurate results. And I don't really have any particular evidence to support that reaction. But the idea that a potentially pathogenic organism like E. coli can take out residence in an internal organism, even a potentially tolerogenic organism like the placenta, without any inflammatory reaction kind of makes me a little bit uneasy. So what are reasons why we might treat the placental microbiome with some caution still? So firstly, the technology for the detection of organisms of the microbiota is really very, very good. You might only expect 0.002 milligrams of bacterial DNA per gram of placental tissue, which means that there is a very low biomass present. If these organisms do constitute a microbiome, there are very, very few of them. And that increases the chance that the bacterial sequences that you are detecting actually come from somewhere other than the placenta. Also, it's really hard to collect a completely sterile sample in a clinical setting. So when we talk about a sterile sample, when we're trying to culture something, the kind of sterility that we're requiring is not quite the same as you would need for a sample where you are basically sequencing the whole sample. So in addition to that, there's what's known as the ketome effect, the kitome effect, which is basically contamination of reagents, of extraction kits and fluids. It's referred to as the ketome, and it becomes very important when the bacterial biomass is expected to be low in the sample. So if the kits that you are using contain bacterial DNA, and the sample that you're using contains very, very little bacterial DNA, then what you're picking up might be what came with the kit. Similarly, the splashome, which refers to the possibility of splashes from one well to another, for example, when preparing samples. Obviously, it doesn't need to be an observable splash. It can be a microscopic splash that results in contamination. And then also several studies, including the AGARD study, shows overlap between the genera described in the placenta and common water and soil genera, which makes the idea of ketomes and splashomes and so on a little bit more, excuse me, a little bit more acceptable. And then the other thing is that it is possible to deliver germ-free animals from non-germ-free mothers. So you can have a normal mouse with a normal microbiome and, no antibiotic exposure. This is a completely normal mouse with a completely normal microbiome, and you can deliver a completely germ-free animal from that mother by cesarean section into a sterile environment and keep them sterile by raising them in that sterile environment. Now, if your microbiome is established in utero, you would think that that would be impossible to do. So you may have a very low biomass of organisms present in what you thought would be a germ-free mouse, but it would be impossible to do if our microbiome was established in utero. I don't have any answers when it comes to this particular component. I think there are more questions than answers, but it's a very interesting area to watch. At the moment, the evidence tends to be kind of balanced. The experts are hotly debating it. I'm probably falling off the fence in the direction of the sterile womb, but equally happy to be convinced otherwise as more evidence becomes available. So while it might yet be unclear exactly how colonization of the human host by our various microbiota starts, it's probably fair to say that you get your biggest dose of microbes soon after you're born. And we realize that this colonization is essential for effective maturation of the immune system. The immune system and the microbiome develop in parallel in early life. So what do you need for your, how do your gut commensals help? So secretion of antimicrobial peptides by neutrophils and intestinal epithelial cells is attributed to gene induction by gut commensals. So if you don't have the gut commensals, you don't generate antimicrobial peptides. The maturation of the Th2 dominance is driven by bacterial interactions. So the switch from your Th2 dominance to a Th1 profile is driven by bacterial interactions, which help to promote barrier function and to regulate Treg and T helper cell balance. Bacterial products and particular species like Clostridia induce functional Tregs in the colonic lamina propria, which seem to protect against immune-related disease and tolerance to promotes tolerance to food antigens. An increased abundance and diversity of Bacteroidetes is associated with improved Th1 responses in neonates. Segmented filamentous bacteria attach and penetrate the epithelium of the terminal ileum, which then induces Th17 cells. Insufficient microbial stimulation results in IgE class switching of mucosal B cells and increases serum IgE levels. Microbiota antigens and metabolites promote plasma cell differentiation in the mucosa and systemically. And then there's this positive feedback loop of IgA secretion, coating and uptake of luminal microbes, processing in the Peyer's patches, and reinforcement of antigen-specific responses. And then IgA as well modulates gut microbiota composition. Again, segmented filamentous bacteria stimulate T-cell independent IgA production in the neonate. And the microbiome remains unstable in humans for about 2 years. So you have this sort of critical window where you can restore or modulate the composition of the microbiota, which can potentially restore immune development. If you have missed that window, if you are an adult and you have a dysregulated microbiome and dysregulated immune responses as a result of that, it's probably not true that you can reestablish normal immune responses by adjusting the microbiome. And so really what I'm trying to say is that you should love your microbiome. You should try not to kill too much of it with antibiotics, and the same applies to your patients. You should treat your microbiome with love and respect because those bugs are keeping you alive. Thank you.
Cool. Thanks very much, Sarah. If anyone has questions, just type them in the chat or raise your hand. But I guess just to kick off, because we've just got a couple of minutes left for the next half. Do you think, what aspect of this do you think is most likely to impact on us in infectious diseases in the future? Is this still a long way off? Isn't this still really kind of basic science, or do you think there's more kind of clinical applications coming around the corner, or is that still many years away?
Well, my personal feeling is that we don't really understand enough about it to be messing with it. I think we are realizing that there are problems associated with dysregulation of your microbiome, but I don't think we're anywhere near understanding how to intervene and how to adjust it. I mean, obviously, there's probiotics and all that sort of stuff, and it's almost certain that they don't do any harm, but who knows if they do any good. The best that we've got in terms of reestablishing your microbiome is the fecal transplant, and that is such a sort of nuclear bomb of an intervention, and I don't think that we are. I don't think that we really understand enough to be causing to be messing around too much with it unless we understand more. That's my feeling.
Yeah, I think that's a very good point. I'm just gonna, Zamor, you've got your hand up. I don't know if you want to ask a question.
Yes, thank you very much for the talk. Perhaps I could comment a little bit from the GIT perspective, because it's also been quite topical from our side. And yes, it is pretty much experimental, scientific, with your animal studies and such. But from a real world perspective, where there is data, in fact, even some of our guidelines have changed. And from an ID point of view, is certainly in the realm of C. diff, particularly with recurrent or prevention of recurrent C. diff. There's definitely data with regards to modulation of the microbiota or the microbiome in that instance. So there is a role for fecal microbiota transplant there. And also in the setting of fulminant C. difficile infection. There certainly is some good data there. But another comment, just where we are getting a little bit more, and I know our rheumatological or rheum colleagues, as well as our endocrines, there's a lot of data coming from your autoimmune diseases, your thyroid, as well as your rheumatologic, in fact, even your metabolic diseases. And there have been some mice studies that have shown the role of the modulation of the microbiome in these instances as well, with some decent results. So there are some, now going into the realm of your randomized controlled trials and such happening. So I think it's very exciting, and probably the next couple of years we might have some data coming out.
Cool, thanks.